Welcome back to the deep dive. Today we are zooming in, really zooming in, on the molecular front line of cancer treatment. If you want to understand how we actually get from promising research to effective personalized immunotherapies,
Well,
well, you have to look at one very specific interaction. It's this microscopic handshake between a cancer peptide and an HLA molecule. Scientists call it the pHLA complex. And this tiny little docking station, it's the entire foundation of our T-cell response.
it absolutely is. And that handshake, that physical structural interaction, it's everything. For successful neoantigen discovery, which is just finding those unique tumor flags for the immune system to attack, we need precise maps of that pHLA complex. But historically, we face this huge bottleneck. Getting high-resolution experimental structures of these things it's incredibly difficult and scarce. And so we have this fundamental knowledge gap. And it's been holding back the design of safer, more precise therapies.
Right. So our mission for this deep dive is to see how computational power is not just easing that bottleneck.
Yeah.
It's completely shattering it. We're looking at a quiet revolution here, one that's driven by AI, by massive data sets, and by these user-friendly platforms. And crucially, all this new structural knowledge is finally letting researchers solve what some people are calling the T-cell quality problem, which is all about making these next-gen treatments safer and ultimately much more effective for you, the patient.
Okay, so let's start with democratization, because structural modeling, it used to be locked away in these very specialized labs, right? But now we're seeing platforms designed to bring this power to, well, any bench scientist. We're talking about something called HLA-Arena 2.0. The key thing here is that it's web accessible, user-friendly, and this is huge. It's a no-code platform.
It's the perfect bridge, really. If you're an experimental biologist, you don't want to be spending all your time trying to write Python scripts. HLA Arena 2.0 is built on a container-based modular system.
Okay, what does that mean in practice?
Modular.
It means users can sort of pick and choose from established tools, like their virtual building blocks, and then assemble these really complex prediction workflows. And you can run these either in the cloud, like in Google Colab, or even locally if you have sensitive data, all without any programming background.
So it's basically an end-to-end management system for finding and prioritizing these neoantigens. What are some of the heavy hitters in that toolkit?
It's a really powerful integration. For that initial prediction step, it includes tools you might have heard of, like PVA tools. Then when you need to actually see the physical structure, it integrates state-of-the-art modeling tools, things like APE-Gen and DINC.
Now, you mentioned something that really caught my eye, a feature that elevates this from just a research tool to something that's clinically relevant, safety. You said it includes something called cross-dome. So for our listeners, when we talk about cross-reactivity risk, what is cross-dome actually doing with that structure to make sure a T-cell doesn't attack a healthy cell by mistake?
That's where the structural analysis is so vital. Crossdome systematically checks your modeled pHLA structure against this huge library of known self-peptides and human proteins.
So it's looking for mimics.
Exactly. Imagine the HLA is a hand holding a unique key, that's the tumor peptide. Crossdome looks to see if the body also has a very similar-looking key, a self-peptide, somewhere in normal tissue. If it finds those potential lookalikes, it flags them so researchers can avoid peptides that carry a risk of causing dangerous, off-target autoimmune effects.
And that structural check.
Yeah.
It's just a huge step up from only matching the sequence, the letters. It lets researchers use predictions of structural stability, of actual binding affinity. It helps them find these really promising immunogenic peptides that, they might have completely overlooked if they just stuck to the old sequence predictors. The system is just getting so much smarter.
Okay, so for making these tools accessible, let's talk about sheer unprecedented scale, because even the best platform relies on its underlying models, and those models need just enormous amounts of data to learn from. The next big frontier is moving past just looking at the letter combinations of a peptide to accurately predicting the structural features of the entire complex, the pHLA and the T-cell receptor together.
That's the challenge, yeah. The sequence-based tools are good, but they hit a ceiling. They just can't see the physics of the interaction. So to break through that ceiling, researchers launched this enormous AI project. They started with a labeled data set from an earlier tool called Big MHC. And that data set already had over 15 million pHLA sequences.
15 million. 15 million. All linked to experimental results telling them if a T-cell reacted or not.
So that's the foundation, 15 million sequences. But how on earth do you turn that astronomical number into structural models without it taking, I don't know, a decade? That sounds like it needs some serious industrial-level computing.
It absolutely does, and they're using two independent, highly optimized modeling approaches at the same time. First is APE-Gen 2.0, which is built
Huh?
specifically for this kind of scalable pHLA modeling. And second, and this is where it gets really, really interesting, they're using a tool called Boltz-2.
Boltz-2. Okay, so we've all heard of tools like AlphaFold 2 that have revolutionized protein prediction. What does Boltz-2 do that makes it better for this specific job, for these 15 million complexes? Why is it outperforming the big general predictors here?
Well, AlphaFold 2 is fantastic for general protein folding, no question. But Boltz-2 is specialized for the biophysics of binding complexes. It has this improved biophysical refinement that's specifically tailored for docking structures like pHLA. And most importantly, it uses a novel AI approach to predict the complex's binding affinity.
How tightly they stick together.
Exactly. And that strength of interaction is a critical signal for how potent a T cell might be. By prioritizing that binding physics in its prediction, Boltz-2 just offers a level of accuracy that the general models can't quite match in this specific area.
So you're throwing two of the best, most specialized modeling tools at the world's largest data set.
That's the plan. They are currently modeling the entire 15 million complex dataset with both APE-Gen 2.0 and Boltz-2. When it's all done, they expect to have around 30 million unique modeled pHLA structures. And this gargantuan dataset, it's going to be the largest ever created for this kind of machine learning. It's the bedrock we need to improve immunogenicity prediction by leaps and bounds.
Okay, so we're getting close to mastering the target, the pHLA complex. Let's shift focus now to the weapon itself, the T cell. Because we've all seen cases where a therapy seems perfect on paper. You know the target is there, you can confirm the patient has tumor-specific T cells, and the therapy still fails. So why would a perfect target be ineffective if the T cell army seems weak?
This is maybe the most fundamental question researchers are tackling now. What actually defines T cell quality? And they investigated this using mathematical modeling specifically, something called ordinary differential equations. It's a way to simulate dynamic systems like T cell populations competing for resources inside the body.
And the system they modeled had a finite carrying capacity, right? There's only so much space, so many resources for T cells in and around a tumor.
Precisely. And what they discovered was that T cell clones vary drastically in their potency. Some are absolute champions, others are, just milling about. And the model revealed this really counterintuitive problem. When these mildly effective tumor-specific T cells dominate the initial response, they take up all that finite carrying capacity. They're occupying all the space.
So these mediocre T cells are essentially clogging the system. They're specific to the tumor, yes, but their killing power is too low to actually clear it, and they're blocking the really potent T cells from getting in and doing their job.
That is the chilling implication. The mild clones are worse than useless. They're actively inhibitor. The model predicted that just removing these abundant but mildly effective clones actually restored the anti-tumor activity.
Wow.
Because it freed up the carrying capacity for the few highly potent clones to expand and wipe out the tumor. It just reinforces this vital principle.
Right.
The T cells we use in therapies have to be selected not just for being antigen-specific, but for being demonstrably highly potent. Quality control is everything.
That's a fascinating and frankly kind of alarming finding. It completely changes how we have to think about therapy design. So if we know we need these super potent T cells, how do researchers make sure they select them? Or even better, how do they design them from scratch? Let's look at this structure-guided pipeline that was developed for acute myeloid leukemia, focusing on a specific neoantigen, TP53Y220C.
Yeah, this project is a beautiful example of tying it all together: structure, design, and patient data. They used a really clever three-step process. Step one was pure structural design. They used a deep learning tool called protein MPNN to redesign the specific binding sites on the T cell receptor, the CDR1, CDR2, and CDR3 loops.
Let's just clarify what those are. The T cell receptor is what recognizes the pHLA complex, but the pHLA is the key.
Yeah.
What are the CDR loops?
Think of the T cell receptor as a hand. The CDR loops are the three specific, intricate fingers it uses to physically grab onto that pHLA complex. So redesigning these loops means optimizing the T cell's grip on its tumor target. Using this tool, protein MPNN, they generated 50,000 candidate TCR sequences, all optimized for that perfect grip on the TP53 neoantigen.
50,000 digitally engineered T cell receptors. That's an incredible scale. So what do they compare them against?
They needed a reality check.
Yeah.
So step two was patient mining. They went and looked at unique TCRs that were derived from actual AML patients using single-cell TCR sequencing data. And then step three was clustering and verification. They used tools like GLI-PH2 to see if their 50,000 synthetic candidates lined up with what the patient's own immune system was doing successfully.
And did they? Did the synthetic design find the same solution that the natural human immune system did? Because that kind of convergence would be a powerful validation.
It was a very powerful moment. The analysis showed this clear convergence. Two of their computational design TCRs shared a common key motif, this essential sequence in the CDR3 loop with a dominant patient cluster.
On Dominic
And this wasn't a small cluster. It contained over 90% of the patient samples who were responding to that neoantigen. It means the computer, just by using structural physics, arrived at the exact same solution that nature prefers.
That gives you immense confidence in the approach, and I see they completed the loop by taking it back to structural modeling one last time.
Yes, exactly. They took 6 of the best designed TCRs and 62 patient TCRs from that dominant cluster and modeled their structures using TCR Model 2. They're now being evaluated with another tool, STAG, to really dig into the predicted binding specificity and the interface similarity. This whole integrated strategy combining design, mining, and validation, it provides this scalable, reproducible pipeline to discover highly potent, personalized TCRs for really tough cancers like AML.
Okay, let's unpack all this for you. We started this deep dive with what was a debilitating structural knowledge gap. And we've seen that gap start to close with user-friendly platforms like HLA-Arena 2.0, which provide built-in safety checks like Crossdome. Then we saw the scale just explode with an AI initiative that's building 30 million structural complexes using specialized tools like Bolt2. And all of this foundational work, this mastery of structure, is now feeding directly into therapeutic strategy, not just for design, but for vital clinical warnings about T-cell quality.
The shift is really from, hoping a sequence works to having real structural confidence in the interaction. These tools are enabling much more confident peptide prioritization because they can assess stability and off-target risk. The entire field is moving toward a much higher safety and efficacy profile. We are, in a way, refining the immune system's vocabulary.
So we'll leave you with this final provocative thought to consider. If these mathematical models are right, and they are grounded in the reality of finite cellular capacity, and they show that selecting rigorously for high potency is necessary for success, even if it means actively removing the majority of a patient's own tumor-specific but mildly effective cells. What revolutionary logistical and procedural changes need to happen in the clinic to actually scale up personalized T-cell therapies that are built on this principle of rigorous exclusion and quality control? It's a challenge that takes us far beyond just simple antigen targeting.